Okay, so why don't we get started here? People can come in as they're going along. It's my pleasure to host today. We're going to hear from the laboratory of Matt Lorincz, both Julien Richard Albert and Matt Lorincz himself. For the trainees out there, I just want to say that I've known Matt for 20 years. We were postdocs together at the Fred Hutchinson Center. And what's really nice is that we've been able to maintain this great friendship over so many years, and things like the CERC network and conferences allow you to see one another very often and catch up on things and keep in touch. So we talk about all the things that are difficult about being a PI. There's a lot of really nice benefits about being a PI, and it's being able to keep in contact with great people like Matt, and just showing what a great person he is. He's collaborating with so many people around the world, top people in the area of epigenetics. They all enjoy working with them. And you'll see, if you look, do a PubMed on Matt, he's got papers in all the top journals. He had a recent collaborative paper come out in Science, and papers in Nature Genetics. I think he's going to tell us about a Nature Genetics paper that just came out of his laboratory today. But we'll get to Matt in a second. We're going to start with a talk first by Julien Richard Albert, who just finished up his PhD in Matt's laboratory. Julien is originally from Montreal. He did his Bachelor of Science at Concordia before moving out to UBC to work with Matt, and just recently finished up. So congratulations, Dr. Albert, on completing your PhD. He's now living in Paris. He's at the Institut Jacques at the Université Paris, where he's working with Maxime Greenberg and looking at the relationship between DNA methylation and the polycomb group proteins. But first, we're going to hear about the work that he did during his PhD. And the title of his talk is How I Met Your Mother, Maternally Transmitted DNMT3A Regulates Paternal Gene Expression in the Early Mouse Embryo. So take it away, Julien.
Okay, thanks so much for the kind introduction, Jeff. And I wanted to start off by first thanking the organizers of this event, including Damini and Tony, for putting on this nice event. And just more in general, the current and past organizers of all SSHRC meetings. They're a really good way of showcasing great Canadian research. I've met tons of great people at all your conferences, and they've really been a highlight of my PhD. So thank you for letting me present at one of your meetings. And I put this picture on of the Eiffel Tower down here, just to say that, as Jeff mentioned, I'm in Maxime Greenberg's lab now in the COVID-ridden Paris. And I also included this little bird because Max, as is like sort of hazing, he forces all of us to use Twitter. And so if you can please give me a couple of followers, he'll give me wine if I get like a certain amount of followers later tonight. So check me out on Twitter. But today I'll be talking to you about a recently published project that I worked on in Matt's lab at the University of British Columbia. And in that project, I studied what happens on a molecular level to one of these itty bitty tiny sperm after it fuses with one of these huge eggs. Can you see my mouse, by the way? I'm just gonna assume you can. Okay, thanks. And so, yeah, so one after. Okay, and this natural process of fertilization is really interesting, both for developmental biologists and epigeneticists, because you're essentially taking two highly differentiated and unipotent cells and then creating the one cell zygote, which can then divide and become any cell type of your body. So specifically in Matt's lab, we use isogenic mouse strains to study epigenetic mark dynamics in the developing mouse. And we use isogenic mice strains because within any given strain, all individuals are genetically identical to one another. So they're all clones of one another, which really, it still blows my mind to this day. And we have at least 18 such clonal mouse strains in existence. And by using these mice, work in Matt's lab and others all around the world have shown that sperm and oocytes have entirely distinct epigenomes. And so this includes unique DNA methylation profiles as well as transcriptomes. And the functional relevance of having unique chromatin was known long before we knew its molecular basis. So 35 years ago, Azim Surani and his colleagues asked the basic question: if the genetic sequence of your mom and your dad are identical, do we even need a mother and a father for conception? And so in this seminal study, they used micromanipulators to remove the maternal genome from the egg, and then perform 3 key experiments, reconstituting these with a maternal genome from another egg, as well as a paternally inherited genome from a sperm, which amazingly creates embryos that develop to term. And they also reconstituted them with 2 maternally inherited genomes or 2 paternally inherited genomes, both of which failed to develop. So this demonstrates that despite being identical in genetic sequence, you still need a mom and a dad for normal development. And our best explanation for why these embryos fail to develop is due to the requirement of maintenance of parent-specific DNA methylation, specifically at imprinted regions, of which there are about a dozen in human and mouse. So fast forward to today, and here is a summary of DNA methylation dynamics in the mouse. And so, as I mentioned, the male and female establish differential levels of methylation during gametogenesis, and Matt will talk to you about this during his talk. For the rest of my talk, I'll focus on this stage, asking the question, what happens to this unique methylation following fertilization? And so studying methylation or parental methylation in the gametes is relatively easy because you can simply take your sperm cells, put them in one tube, take your eggs, put them in another tube, conduct your experiments like whole genome bisulfite sequencing, and then compare your 2 data sets. The problem with studying 2 cell embryos and beyond is that each cell contains 1 copy of each of your parental chromosomes. And so in order to recapture maternal and paternal methylation, you need allele-specific approaches. And the classical allele-specific approaches simply use microscopy to visualize global levels of DNA methylation in the zygote. And so if you stain DNA with an anti-5 methylcytosine antibody, which in this case glows white if there's a lot of DNA methylation, you can see that in the zygote, before the parental pronuclei fuse, so they're still distinct, before they fuse, you can measure differential methylation. And so you can appreciate that even before the cell divides, the paternal genome undergoes preferential loss of methylation. Unfortunately, in the 2 cell and 4 cell and beyond, you can no longer trace the inheritance of methylation because, like I said, there's 1 copy of each parental chromosome in each cell, and let alone measure methylation at specific loci that you're interested in. Because we know that parental imprints are maintained during development and that global levels of paternal methylation are lost following fertilization, we don't really know what happens at other sequences. So I circumvented these limitations by taking advantage of F1 hybrids, which are generated by crossing distinct mouse strains, as well as high throughput sequencing approaches. And so I don't have time to go into details, but my colleague Tesuku from Japan and I developed this fancy pipeline we call MIA, which analyzes DNA methylation, ChIP-seq, and RNA-seq data with allele-specific resolution. And how it works is you input into the pipeline the known genetic variants that are different between your 2 parental isogenic mice. And then the program uses these genetic variants as genomic landmarks to delineate whether a read originated from the maternal allele or the paternal allele of the F1 hybrid. And then it separates those maternal paternal reads into separate files, and then you can compare them. And so I had this new fancy pipeline, and the first thing I applied it to was early mouse embryonic WGBS data. And for the rest of the talk, I'll be focusing only on the paternal allele. So here is my fancy plot where each line represents a 2 kb bin. And where that 2 kb lands on the y-axis corresponds to the average CpG methylation within that 2 kb bin. So as the lines load, you can see that the pattern of methylation is generally in line with the dogma. So this is a nice orthogonal way of showing essentially the same thing. But the advantage of whole genome bisulfite is that it's really high resolution. And so if you want to make this graph interactive, you can actually go through the different bins and identify those that show interesting patterns. And so I highlight this one because we're interested, as I said, in the inheritance of methylation. So comparing sperm to 2-cell, unlike the rest of the genome that undergoes loss of DNA methylation, this region shows a gain of methylation. And when I dug deeper into what these regions are, I noticed that they were enriched for CpG island promoters, right? And so in a more focused analysis, what I'm about to show you is only sperm versus paternal 2-cell at CpG island promoters. So here each dot is a CpG island promoter. And you can see that while most CGIs remain hypomethylated or unmethylated following fertilization, a subset strikingly show like this nice gain of methylation. And so this is completely counter to the dogma that the paternal genome undergoes this widespread loss of methylation. So we were really interested in this. Here's what it looks like at an individual gene level. So I'll just walk you through really quickly. In blue is the CGI promoter that I've highlighted. And then for each WGBS data set, I'm showing the percent methylation as a bar from 0 to 100%. And then below these bars, I'm simply showing the location of each CpG for which we had sufficient allele-specific coverage to call DNA methylation. So that those are highlighted in yellow. So you can see in sperm, the CG island promoter is unmethylated. And then following fertilization, unlike the rest of the genome, you see this nice gain of methylation across the entire CG island promoter. And so the fact that like 30 or 50 individual CpGs show this gain in methylation really gave us confidence that this was a real effect. And to validate this effect further, we integrated K4 trimethyltrypseq because it was shown biochemically that this histone modification impedes de novo methylation. So if you're K4 trimethylated, you don't get DNA methylation. And so if you see a gain in DNA methylation, you expect to see a loss of K4 trimethylation at the same stage. And that's exactly what we see. So this got us excited. And the first question we asked was, what happens to this methylation? So here we're at the 2-cell stage. And in the previous graphs, I showed you that DNA methylation keeps going down to the inner cell mass stage. So what happens to these loci later in development to the blastocyst stage? Here's just a quick summary of what we found, where if we integrate F1 hybrid blastocyst data, if we take out methylation from the paternal allele, you see that while about 25% of our regions of interest lose the methylation, so that gain was only transient, a nice portion of them maintain intermediate levels of DNA methylation to the blastocyst stage, where global methylation levels are super low. And what's really cool, in collaboration with Hisato Kobayashi, he did WGBS on those uniparental embryos that I introduced to you earlier. And we show that this effect really only happens when you have at least one paternal genome, right? So this gave us further evidence that this was a real effect. And not only that, but the fact that this methylation is maintained to somewhat of an extent to the blastocyst made us think that it might have a biological role. And the first way to determine what its biological role was is we had to identify the factor that mediates the gain of methylation on the paternal genome. And in mouse, there's 2 flavors of de novo DNA methyltransferases, 3A and 3B. And we already know from previous studies that 3A is highly expressed in the oocyte and interestingly hangs around after fertilization in the zygote and the 2-cell, whereas 3B is just not expressed at all. So we collaborated with Hiro Sasaki's lab, who developed and still has conditional knockout mice, where they removed DNMT3A specifically in the developing oocyte, generating oocytes depleted of DNMT3A. And importantly, when you fertilize these oocytes, the resulting embryos fail to develop. And so using these oocytes, wild type with the DNMT3A or DNMT3 maternal knockout, they inseminated these with wild type sperm from a distinct mouse strain in order for me to apply MIA to the whole genome bisulfite sequencing data that they generated from these 2-cell embryos and pull out the paternal specific methylation. And thankfully, in the wild type cells, we still measure the gain of paternal DNA methylation following fertilization. So this just recapitulates what we saw previously in our discovery data set. And amazingly, or I really think this is amazing, when you don't have maternal DNMT3A, you no longer see the gain of methylation on the paternal genome following fertilization. And here are our 2 favorite genes again, showing nicely the gain of methylation in wild type 2 cells, and then the lack of a gain of methylation in the DNMT3A maternal knockout cells. And while our sequencing wasn't as deep as the discovery data set, so we capturing fewer CPGs, it was still a robust finding, I think. So this was really cool. We identified the factor that mediates the paternal methylation acquisition. What's even better is now we have a system where this gain no longer happens. So we can ask what happened, what's going on in these cells that don't show the gain. And so our collaborators are awesome. They performed the RNA-seq on 2-cell, 4-cell, and ICM cells, wild type, maternal knockout. And here's just the summary of all of the data, where I'm showing you the differential gene expression analysis of all genes and this is from the paternal allele, so paternal allele expression, where I highlight genes in color that are differentially expressed. So over 2-fold upregulation or downregulation. And then I highlight, again, in blue, those that we found previous evidence for DNA methylation acquisition. And so what's really striking is, let's say in the 4-cell stage, of the top 6 upregulated genes from the paternal allele, 4 of them had previously shown a gain in methylation at the CGI promoter. And so what we can say is that this gain in methylation is a brand new dynamic of DNA methylation on the paternal genome in the very early embryo, and that this new methylation that we've kind of uncovered seems to be important for the regulation of genes on the paternal allele. So that's pretty much the summary. That's all I have to say. There's still some major unanswered questions, but we're happy with this project. It was recently accepted after a nice peer review. So we're very happy about that. And yeah, so this was like the first application of the fancy pipeline to answer a biological question. And we really owe it all to our awesome Japanese collaborators. So I would just like to acknowledge them. So the researchers in Hiroyuki Sasaki's lab, who did the maternal 3A knockout experiments, as well as Hisato for the uniparental WGBS, Tesuku for helping with the pipeline development, as well as Matt for being an awesome PI and mentor, as well as Aaron and Julie, who held my hand just throughout my entire PhD. And my supervisory committee for having my back, as well as these shirk meetings, as I mentioned earlier. They're my favorite meetings to go to. So I hope they keep happening soon. And yeah, with that, thank you. Happy to take your questions.
Okay, great. Thank you, Julien. It was a beautiful talk. So I remind everyone that questions can be sent to me through the chat window. And I will start with the first question, Julien. So it's really nice the way you showed that this DNA methylation was occurring specifically at the 43 genes, and it was coming from the maternal DNMT3A. But do you think that there's something that's recruiting the DNMT3A specifically to those genes, or do you think that somehow it's excluded from all of the other genes? Is it a recruitment or a blocking that's happening?
Yeah, that's a great question. And we've been struggling to answer that question for a long time now. Um, there's no clear answer other than it's probably a little bit of both, of recruitment and blocking. And on the recruitment side, we think that there's likely a sequence-specific factor, and it's probably a Crab ZFP, which we know comes in and recruits, binds to sequence-specific factors, and recruits machinery that deposits methylation, DNA methylation, as well as canine trimethylation. And I think it's Crab ZFPs because I didn't have time to show it today, but these regions also gain canine trimethyl at the same stage. Yeah, and in terms of excluding, so it's really only these few loci that show the gain. So it's a bit harder to explain the excluding because you'd have to really exclude everything except for these two. But what we notice is that K4 trimethyl, which is anti-correlated with DNA methylation, is maintained on the paternal allele following fertilization at some genes. So at those, the maternal DNMT3A can't act on.
Yeah. Great, thank you. So I've got a question from Martin Hirsch, which is, does the paternal allele take on the maternal H3K4 trimethyl state at these CGIs, or does this originate from the paternal allele, i.e. through the sperm?
Yeah, so we investigated whether there were any histone modifications inherited from the sperm that might mediate this effect. And of all the ones that exist, we didn't find anything unique to these sites. But what's really cool is what's happening on the maternal genome, which I also didn't present today. And this is going to be, I think, an entirely new project or an entirely separate project in MassLab because there's a lot of interesting things happening. The maternal allele at these sites, well, I don't know, I can go on, but they're not necessarily K4 trimarked is what I'll say. A lot of these are already repressed and not by DNA methylation, but by polycomb. So K27 trimethylation, which also seems to impede de novo methyltransferase at CpG rich regions.
Okay, great. So the next question comes from Wendy Dean. And so Julien, can you comment on why the androgenetic embryos have so little DNA methylation compared to the normal?
The androgenetic, so those are the mother. Oh yeah. This was we got this question from someone else as well. And I dug into it a little bit. So I compared androgenetic DNA methylation. Wait, oh I get mixed up. I haven't touched this in a while. Androgenetic is motherless. So it's made with two paternally inherited genomes. Okay, yeah. So I compared the androgenetic whole genome bisulfite versus the paternal allele whole genome bisulfite. And we do indeed see, it's not only at these regions, it's genome-wide, we see lower methylation levels. We don't have a great explanation for why. We think it's interesting. We're not sure why. It could be that the extra embryonic cells, we don't know whether the extra embryonic cells proliferated more in these blastocysts, but we know based on the phenotype of the embryos that go on a bit farther that they have bigger placenta. And we know that these extra embryonic cells tend to have lower global methylation levels. So it could be a cell type specific thing we're measuring, but we don't know.
Okay, so last question coming from Michael Johnson. You find that paternal CpG island methylation promotes the expression of certain paternal alleles. Are the maternal alleles of these genes also expressed? And if so, is there a detrimental effect if expression of the maternal allele is similarly elevated?
Right. So of the genes that I highlighted as differentially expressed, from memory, I don't think any of the maternal ones are upregulated in the knockout as well. So this is really a paternal allele specific effect. But what's interesting is that at some of these CPG islands, they are just normally expressed in oocytes. So these genes, for whatever reason, like they can be on at that stage and the embryo is fine. So we have a little bit of everything.
Okay, so there is a follow-up question from Wendy Dean. She wants to know how you distinguish DNA methylation from hydroxymethylation.
Yeah, so we can't. This was all, at least all the stuff I presented as like our data was all bisulfite converted DNA, which includes hydroxymethylation. So we know some of these CGLM promoters lose their methylation by the inner cell mass stage, and those are probably have higher hydroxymethylation. Yeah.
Okay. So while you're voting, I'm going to introduce our next speaker, who is Matt Lorincz. And as I said before, Matt and I have known each other for a long time. Matt is originally from the States, and he did his PhD studies at Stanford University before moving to the Fred Hutchinson Center in Seattle for carrying out his postdoctoral work. And there he was working with Mark Rudin, and my wife was across the bench from him in the laboratory there. So that's how we got to know each other quite well over that time there. But of course, we were both very much interested in epigenetics. In 2005, he moved to UBC and has progressed through the ranks and is a full professor there right now. Matt, over his career, he's had a long-standing interest in the interplay between DNA methylation and histone modifications, both in early development as well as gamete development. And what he's done to contribute to the field over the years is really helped us understand how the DNA methylation is regulating the expression of retrotransposons and retroviruses, which are endogenous to the genome. And of course, these retrotransposons and retroviruses get expressed at certain times during the development, but then you need to shut them down. And so Matt has really done an extraordinary job over the years, publishing many beautiful papers, really helping us understand how the DNA methylation is regulating the expression of these different factors. And so today, I think that he's probably going to tell us about this recent beautiful paper that he had in Nature Genetics, looking at the role of NSD1 and H3K36 which is an H3K36 methyltransferase. So he was looking to see what's the interplay between this epigenetic mark and the depositing of DNA methylation across the genome, but more specifically at imprinted genes. So with that, Matt, I turn the floor over to you.
Great. Thank you so much, Jeff, for that kind introduction.
Tony, can we pull off the poll now and
yes,
get up on the screen? Perfect.
And great, perfect. Okay, so again, thank you, Jeff, very much for that kind introduction. Also to Marco and Martin for the invitation to present in this webinar. I think it's great that you all are doing this. Also, thanks to Tony and all the organizers for putting this together. And I look forward to the upcoming talks and next year's meetings, which hopefully we can have in person.
Hey, Matt, your presentation is in presentation mode. That's what we're seeing it. Yeah.
Sorry, hold on one second. How about that?
Perfect.
Just, okay. So great. So today I'm going to tell you about work in the lab that really is a follow-on to much of what we've been doing the last few years, aimed at understanding the interplay between DNA methylation, histone modifications, and transcription. And the work I'm going to describe was carried out for the most part by two very talented senior researchers in the lab, Julie Brindamore, who's a research associate, and Kenjiro Shirane, who recently finished his postdoc, his Banting postdoc in the lab, and has started his own lab at Kyushu University in Japan. Okay, so you just heard from Julien about DNMT3A, the de novo methyltransferase, and to remind you, it's one of 2 de novo methyltransferases that are widespread in mammalian genomes. And I have to point out there's a third recently discovered from Deborah Borkus's lab, DNMT3C, which interestingly enough has the catalytic domain and activity that 3A and 3B do, an ADD domain, but lacks the PWWP domain. I'll be talking about PWWP and ADD domains of these enzymes a little later in the talk. And of course, there's also another de novo methyltransferase family member, a paralog DNMT3L, which lacks catalytic activity, but stimulates the activity of DNMT3A. Now, for all of these de novo methyltransferases, the CG dinucleotide is the favored substrate. And I should also mention here that DNMT3C, although it methylates CGs, is targeted specifically in the male germline to young endogenous retroviruses and retrotransposons to mediate their methylation. Okay, so of course, the CG dinucleotide is a perfect substrate for a true epigenetic mark, and that the complementary CG can be methylated after DNA replication, and this is precisely the role of the maintenance methyltransferase DNMT1. So you saw this slide in Julien's talk. To remind you now, for the work I'll be presenting, we're looking earlier in development, specifically in the germ cells themselves. And so in terms of the dynamics of DNA methylation, one of the reasons we're so interested in the germline is that it undergoes really massive waves of de novo methylation, really reflecting dramatic changes in chromatin and transcriptional programs. And so after germ cells emerge around embryonic day 7, they proliferate and migrate to the genital ridge, and DNA methylation is being reduced during this time and reaches a low point around embryonic day 13.5. And this is the stage in development, we think, where DNA methylation is at its lowest levels of any other cell type in mammals. So shortly thereafter, in the male germline in prospermatogonia, as I'll show you, de novo methylation takes place really dramatically, reaching very high levels by birth. Whereas in the female germline, de novo methylation is delayed until after birth and takes place in growing oocytes. And I'm going to show you data for growing oocytes as well. Now, this methylation in growing oocytes is also dramatic, but doesn't quite reach the levels we see in the male germline. And if we look at this in terms of sort of sheer numbers, essentially the genome goes from being about 10% or less methylated at E13.5 to about 40% of CGs methylated in the female germline and about over 80% of CGs methylated in the male germline. So this translates into on the order of 8 million methylated CGs in the female and 16 million in the male. Now, a curiosity of methylation in these germ cells is that CPG islands show the opposite pattern, with a significantly greater number of CPG islands methylated in oocytes than in sperm. And we can come back and talk about this at the end. So a number of years ago, Kaneda and colleagues showed that in the female germline, de novo methylation is dependent entirely upon DNMT3L and its DNMT3A and its cofactor DNMT3L. And you heard from Julien that maternal DNMT3A actually is inherited in the zygote and then engages some methylation of the paternal genome. Now, what's quite interesting is if we look at the male germline now in PSGs, somewhat surprisingly, I think, considering the difference in the levels of DNA methylation, this de novo methylation is dependent on the exact same factors, DNMT3A and 3L. So this suggests that the difference in DNA methylation isn't dependent simply on the complement of de novo methyltransferases and cofactors, at least DNMT3L expressed. And so to give you a sense of what this DNA methylation looks like genome-wide, or broadly in the genome, we're looking at a region on chromosome 1 here, and following the DNA methylation dynamics through female and male germline development. So you can see that very low levels of DNA methylation at E13.5, and it's quite similar between the male and female germlines. But in the male germline, we see this massive gain in DNA methylation such that in sperm, at this level of resolution, it looks like the whole genome is methylated. But in fact, only those regions that are marked with H3K4 methylation, which reflect promoter regions, lack DNA methylation in sperm. Whereas in the female germline, we see this more block-like pattern of DNA methylation, which I'll describe in greater detail as I get into the talk. But this really reflects this very significant difference. It illustrates a difference in methylation levels in oocyte and sperm. Okay, so what is the evidence for an interplay between histone modifications and de novo DNA methylation? So as I mentioned earlier, de novo methyltransferases have, in addition to their catalytic domain, PWWP and ADD domains. And over a decade ago, it was shown by Steen Oy and Tim Bester's lab that the ADD domain of DNMT3A actually senses the methylation state of H3 lysine 4, such that when lysine 4 is methylated.
Sorry, Matt, to interrupt you. You have like a Sophis antivirus warning showing up on your screen.
It's not on my screen. Sorry about that.
Yeah, no worries.
Okay. Are we better?
Perfect. Sorry.
Okay. So right. So the ADD domain senses the H3K4 methylation state such that the DNMT3A, and in the context of the tetramer with DNMT3L, does not methylate associated DNA when H3K4 is methylated. And this is essentially why we see, at least in part, CpG island promoters being in the unmethylated state because they're marked with H3K4 trimethylation. Now, interestingly enough, other studies focusing on the PWWP domain have shown that this particular domain mediates the opposite effect in the sense that it senses the lysine 36 methylation state and will specifically recognize and promote DNA methylation when H3K36 is methylated. And through a number of studies by a number of groups, the mechanism that's emerged here is that the H3K36 methyltransferase set D2 via its SRI domain recognizes the elongation competent form of RNA polymerase 2, associating with the RNA pol2 as it translocates along genes that are being transcribed, laying down the H3K36 trimethyl mark. And this is recognized by the PWWP domain of DNMT3 in the context of what I'll be showing you, 3A and 3L, leading to efficient de novo methylation of transcription units or gene bodies. So, as I mentioned earlier, de novo methylation in the female germline takes place in growing oocytes. It's dependent on DNMT3A and 3L. And one of the beauties of studying germ cells during the stages when de novo methylation takes place is that they are not dividing. So in the case of the female germline, oocytes are arrested in the diplotene stage of prophase 1. So we essentially don't need to worry about DNA replication or the role of maintenance DNA methylation in our studies of the dynamics of DNA methylation. So to get at this question of whether DNA methylation in oocytes is guided by K36 trimethylation, Julie took advantage of the low input ChIP-seq method that she developed several years ago to carry out a series of ChIP-seq experiments on oocytes using 200 oocytes, looking at a number of histone marks, including K36 trimethylation. And what she found essentially is that all genes that have K36 trimethylation in the gene body, which actually does reflect transcriptionally active genes, are highly methylated in GBOs. And if we look at an example of one of these genes, so this is the PIK3C3 gene, we see that this gene is transcribed in oocytes, and then this broad domain of K36 trimethylation extending throughout the length of the gene body, and DNA methylation being gained between the non-growing oocyte and GVO stages, such that we see this block of very high levels of DNA methylation beautifully corresponding to the enrichment of K36 trimethylation in this gene. So actively transcribed regions in mouse oocytes are de novo methylated and enriched for K36 trimethylation. And we next wanted to ask whether de novo methylation in oocytes is indeed guided by K36 trimethylation. And to do this, we needed to knock out the enzyme that's known to deposit this mark, which I mentioned earlier, which is SETD2, the only lysine 36 methyltransferase that's capable of trimethylating this residue in mammals. And so we set about doing this. This was a very close collaboration with Louis Lefebvre, my colleague here at UBC, using a ZP3 Cre deleter of SETD2 in the female germline. And shortly through this project, we learned that a colleague in China, Wei Zi, who's at Tsinghua University, was carrying out very similar experiments using GDF9, another Cre deleter for oocytes. And so we engaged in a collaboration with Wei Zi and went on to show that, went on to study the role of SETD2 in oocytes. So essentially in the SETD2 knockout oocytes, we see complete loss of K36 trimethylation as expected, and also total loss of K36 trimethylation in gene bodies. And again, this would be in actively transcribed genes. When we actually went and looked at DNA methylation, what we find is that those regions that are marked with K36 trimethylation, and again, highly methylated in control oocytes, show us essentially a complete loss of de novo methylation in the SETD2 knockout, so suggesting that indeed de novo methylation in the female germ cells is guided by and dependent upon K36 trimethylation deposited by SETD2. So if we look at a genome browser screenshot here, you can see the loss of loss of K36 methylation down to background levels, and loss of these blocks of DNA methylation that we see in gene bodies. And that's essentially replaced by this spurious de novo methylation, almost as if the DNMT3A doesn't know where to go in the genome, and we see a sort of ectopic gain of DNA methylation throughout the genome, mediated by DNMT3A. So we also wanted to look whether this DNA methylation plays a role in, whether SETD2 plays a role, I should say, in DNA methylation of gametic DMRs. So specifically of maternally imprinted genes in FGOs. There are about 20 of these in the mouse. And so if we look at a number of them between the non-growing oocyte and fully grown oocyte stages, so these are control oocytes, we see this clear gain of de novo methylation as expected. But in the SETD2 conditional knockout, this gain of de novo methylation entirely fails. So that suggests that SETD2 is indeed required for DNA methylation of GDMRs. So to give you an example of one of these, here's the impact gene, one of these maternal imprinted genes. The gametic DMR is shown in this peach color here. And what you can clearly see is low levels of DNA methylation in the non-growing oocyte and a block of DNA methylation in the fully grown oocyte, corresponding to the gene and downstream regions. But as you'll also notice here, the DNA methylation extends well upstream of the canonical transcription start site. Now, when we look at the K36 trimethylation, you see that it also extends upstream of the start site. And in fact, here as well, the DNA methylation and K36 methylation correspond quite nicely. But this K36 methylation is lost in the SETD2 knockout, and the DNA methylation levels again are dramatically reduced in the SETD2 knockout. Now, in an independent study, we wanted to ask what was the basis of this K36 methylation and DNA methylation extending upstream of canonical promoters, which is actually quite abundant in the oocyte. And it turns out that there are a number of transcripts in oocytes that initiate in long terminal repeats, so these parasitic retrotransposons. And in the case of the impact gene, there is an LTR well upstream of the CG island gametic DMR and promoter of this gene. And it's in fact transcription initiating in this LTR that generates this domain of K36 methylation and indeed de novo DNA methylation over the CPG island embedded in the GDMR and the GDMR itself, which is what's required for establishment of the imprint of this gene and several other genes in the mouse genome. And this was a study published last year. Nature Communications. Okay, so to summarize then, in growing oocytes, de novo methylation occurs in actively transcribed regions that are marked with K36 trimethylation. This is dependent on the lysine methyltransferase SETD2, and we think this is what explains this block-like structure of DNA methylation that we see in oocytes. This is really just showing us where transcription is taking place in the growing oocyte. And the regions that are methylated include the gametic DMRs, which are embedded in these transcription units. Okay, so now I want to switch gears here for the rest of the talk and focus on what's happening in the male germline. And again, this is work done by Kenjiro Shirane when he was a postdoc in the lab. So as I mentioned earlier, de novo methylation in male germline takes place before birth in prospermatogonia. And again, these cells, in this case, are mitotically arrested, so we don't need to worry about maintenance DNA methylation in the classical sense. And this DNA methylation is dependent, just like in oocytes, on DNMT3A and 3L. So an obvious question to ask is whether SETD2 is required for de novo methylation in the male germline as it is in the female germline. And Kenjiro went about doing this by generating knockouts of SETD2 using the TNAP pre-deleter line, which leads to deletion or excision of the region of interest at, beginning around embryonic day 9.5, well before de novo methylation begins in the male germline. And he did this with an OCT4 GFP so that he could sort and analyze, he could purify prospermatogonia and analyze them by ChIP-seq, RNA-seq, and whole genome bisulfite sequencing. And so, progressing here. Okay, so what he found is that, not surprisingly, in the SETD2 knockout, so focusing at a region here, here's the SFSWAP gene. What we see in the SETD2 knockout at embryonic day 16.5 is a essentially complete loss of H3K36 trimethylation as expected. But surprisingly, if we look at DNA methylation, so in this locus, as expected, we see gain of DNA methylation between E13.5 and E16.5 in control E16.5 PSGs. But in the SETD2 knockout PSGs, we see really no change in DNA methylation whatsoever. In other words, it seems that SETD2 is entirely dispensable for de novo methylation in the male germline. And indeed, that's exactly what we see when we look genome-wide, very different than the picture in the female germline. So when we compare SETD2 control to SETD2 knockout, E16.5 PSG, we see no change in DNA methylation, regardless of whether the region was marked with K36 trimethylation in controlled prospermatogonia. So SETD2 is dispensable for the global wave of DNA methylation that takes place in the male germline. So this then led us to revisit the question and ask, might there be a different chromatin mark that's directing de novo methylation in the male germline? And we turned to a related mark, and that is H3K36 dimethylation, because it was shown a number of years ago by Dylon and colleagues that the PWWP domain of DNMT3A not only interacts with H3K36 trimethylation, but also H3K36 dimethylation. More recently, Ducatz and colleagues showed that this is also true. So the DNMT3A recognizes H3K36 di and tri, and this recognition is entirely ablated by introducing a mutation in a residue within the PWWP domain. Now, complicating matters for us is the fact that while there's only one K36 trimethyltransferase, there are 4 enzymes that deposit H3K36 dimethylation in mammals. And so we first wanted to ask, well, are de novo methylated regions actually marked in prospermatogonia by K36 dimethylation? And to answer this question, Kenjiro generated ULI ChIP-seq now for this mark, H3K36 dimethylation. And what you can clearly see here, if we look between E13.5 and E16.5 in control male germ cells, we see again this dramatic gain of DNA methylation. And now when we integrate the K36 dimethylation data, you see this beautiful correlation between de novo methylation and K36 dimethylation. They almost look like replicates of the same mark, but one's DNA methylation, the other K36 dimethylation. And this is quite different than the distribution of K36 trimethylation, which at this resolution looks much more punctate, but simply reflects the presence of K36 trimethylation in transcription units. So if we look genome-wide in 10 kb bins, we again see this very strong correlation between H3K36 dimethylation and regions that are gaining DNA methylation between E13.5 and E16.5. Okay, so this suggests that K36 dimethylation may be responsible for directing DNA methylation in the male germline. So this brings up the question, which K36 methyltransferase and is H3K36 dimethylation actually required for de novo methylation? Now, Unfortunately, all 4 of the K36 dimethyltransferases are expressed in the male germline at the stage when de novo methylation takes place between E15.5, after E15.5. But only 2 of these K36 methyltransferases had been shown previously in somatic cells to exert a sort of global effect on K36 dimethylation. And work from a number of groups have shown in both humans and mice that DNA methylations are reduced, broadly reduced, specifically in the absence or a decrease in NSD1. So in carcinomas harboring loss of function mutations in NSD1, this is from Nada Javato's lab, there a dramatic decrease in DNA methylation was observed. In Soto syndrome patients who have loss of function mutations in NSD1, this is from Rosanna Wexberg's lab at the University of Toronto, and in NSD1 knockout murine embryonic stem cells, so Jacques Majewski's lab, also at McGill, all showing reduced levels of DNA methylation, led us to focus on NSD1 in the male germline. And so to get at the role of NSD1, we obtained a conditional knockout allele from Pierre Chambon's group in France and carried out similar deletion experiments as we had done in the female germline, and also for SETD1, SETD2 in the male germline, to knock out NSD1 and look in E16.5 if there's a consequence for de novo methylation in these cells. And so again, we use OCT4-GFP, sort the GFP positive proschromatogona, and ask by chip and whole genome bisulfite sequencing, and also looking at the expression by RNA-seq, what the consequences are for knockout of NSD1. And so what we were very happy to see is that in biological replicates looking genome-wide at DNA methylation, we see a dramatic decrease in DNA methylation in the NSD1 knockout. And interestingly enough, this decrease is tempered in regions of the genome that are marked with both H3 K36 di and trimethylation. Although they're decreased, it's not to the extent that we see genome-wide. And in fact, if we integrate K36 dimethylation into the picture, we see quite strikingly that it's precisely the regions losing K36 dimethylation that show the most dramatic loss in DNA methylation, whereas regions showing residual K36 dimethylation are still de novo methylated. So what this translates into is that about 25% of the genome shows a reduction in K36 dimethylation, and about 33% of CpGs analyzed show a decrease in DNA methylation of greater than 20%, indicating that NSD1 is indeed really critical for de novo methylation of large regions in the male during male germline development. So as we did in the female germline, we also asked whether the 3 imprinted genes in male germ cells are dependent, in this case, on NSD1. Looking at the dynamics of this de novo methylation of these imprinted genes, we see that they reach very high levels of methylation at P0, so around birth. And so we looked by whole genome bisulfite sequencing at the consequences of NSD1 knockout for these 3 genes. And indeed, we see that all 3 paternally imprinted genes show a decrease in DNA methylation, with H19 being particularly sensitive to the loss of NSD1 in terms of the establishment of methylation at this GDMR. So finally, we noticed that when we looked at just a genome browser and integrated H3K27 trimethylation analysis, we found that the region showing the greatest decrease in de novo methylation in the NSD1 knockout nicely corresponded to regions that were marked with H3K27 trimethylation. So you can see an example of this in this genome browser screenshot. Shown in salmon here are regions that are marked with H3K27 trimethylation, the tracks at the bottom. So you clearly see that H3K36 dimethylation in these regions is dramatically reduced compared to regions that don't have K27 trimethylation. And as importantly, DNA methylation is also dramatically reduced, specifically in these regions that are marked with K27 trimethylation. And you can see that in a genome-wide view. So if we look at regions that do not have K27 trimethylation, there is a loss of DNA methylation. But if we look at regions that are co-marked with K36 di and K27 trimethylation, the loss of de novo methylation is essentially down to the levels we had before de novo methylation is initiated, as reflected at E13.5 PGCs. So interestingly enough, not only is DNA methylation lost at regions that are losing K36 dimethylation, but if we look at K27 trimethylation itself, and you can see it's the case here in this screenshot, we actually see a concomitant increase in K27 trimethylation in the regions showing a loss of DNA methylation and K36 dimethylation. Now, this increase in K27 trimethylation was particularly of interest to us because when we looked at the RNA-seq data, we found that the vast majority of genes showing a change in expression in the NSD1 knockout are actually showing reduced expression, which is paradoxical given that there's this dramatic loss of DNA methylation. So over 1,300 genes are downregulated. And lo and behold, if we look at the K27 trimethylation status of genes that are downregulated, we see that the vast majority of genes that are downregulated in the NSD1 knockout and losing DNA methylation actually show a gain in H3 K27 trimethylation. Okay, so to summarize then, essentially what I've shown you is that the sexually dimorphic patterns of DNA methylation in the germline, we think are due to distinct profiles of H3 K36 di and trimethylation in prospermatogonia and growing oocytes. So what we see in the female germline is that SETD2 is depositing K36 trimethylation at actively transcribing regions, and this stimulates de novo methylation by DNMT3A between the non-growing and fully grown oocyte stages, such that we see methylation in transcription units of actively transcribing regions. And in the SETD2 knockout, we lose this DNA methylation, but the transcription of many genes is unaffected. I didn't have time to show you that there are a subset of genes that gain K27 trimethylation also in the female germline and are downregulated. But in fact, there's much less K36 dimethylation in the female germline than the male germline. And when we look in the male germline, we see that NSD1 broadly is depositing K36 dimethylation throughout the genome, in addition to SETD2 marking gene bodies of actively transcribed genes. And so between the primordial germ cell and prospermatogonial stages, DNMT3A acting with 3L broadly DNA methylates regions that are marked with K36 di and trimethylation. But in the NSD1 knockout, this de novo methylation doesn't take place, and we lose de novo methylation broadly in the genome, including at regions that are targets of PRC2. And this results paradoxically in a gain of K27 trimethylation and silencing of genes that would otherwise be expressed. And the only regions retaining DNA methylation in the male germline are those that have H3 K36 trimethylation because SETD2 acts independently of NSD1, and a few other regions that are likely H3 K36 dimethylated by one of the other H3 K36 dimethyltransferases that I mentioned. Okay, so with that, let me thank the people that did this quickly. Again, Julie Brenda Moore, working with Aaron Bogus in the lab, and in collaboration with Louis Lefebvre and Wei Zi, worked on the female germline side of the projects that I described. Kenjiro Shirane and our collaborators Takashi Ito and Fumihito Miura generated the data that I presented to you for the male germline, and this work was supported by these funding agencies. And with that, I'll be happy to take any questions.
Thank you, Matt. Great talk. So Martin had the first question. Again, everyone, if you have questions, please send them in through the chat. Martin wanted to ask, so maybe I missed this, but what state of H3K36 dimethyl, oh, sorry. What is the state of, sorry, the screen keeps moving on me as people send questions. What is the state of the H3K36 dimethyl in the maternal genome?
Okay, can you see my screen?
Yes.
Okay, good question, Martin. So K36 dimethylation in the maternal genome. So about 60% of the prospermatogonial genome is marked with K36 di. In the female germline, that number's 30%, with 25% of the oocyte genome marked with K36 tri. But what's quite interesting is if we look at the relationship between K36 tri and di, now in the female germline, so this is in growing oocytes, we see that the vast majority of regions marked with K36 dimethylation in the female germline are actually also marked with K36 tri. And I think you can see that more clearly. It makes more sense if you look in the heat map here, where we're looking at DNA methylation, K36 dimethylation, and K36 trimethylation, looking in control and SETD2 knockout oocytes. And so what you can clearly see as expected is that, as I mentioned earlier, K36 trimethylation is lost in the SETD2 knockout as we expect, as is DNA methylation. But what's quite interesting is that K36 dimethylation is also lost in the SETD2 knockout. And in fact, if we look genome-wide at K36 di and tri in oocytes, we see that the majority of K36 dimethylation is actually dependent upon SETD2. And this is likely because the K36 dimethylation in the female germline is actually deposited by SETD2 at the edges. You can actually see it here in the heat map. At the 5' and 3' edges of actively transcribed genes. So it's just not making it all the way to K36 trimethylation. And so we see these regions as being marked with H3 K36 dimethylation, which I think then begs the question, why is there so little K36 dimethylation beyond what SETD2 is depositing? And we actually don't know the answer to that question, but one interesting observation here is that if we look at the NSD1 locus in oocytes, what we see is that, and I mentioned earlier that LTR elements are particularly active in the female germline, actually in growing oocytes. And we see an example of that in the NSD1 locus, where a long terminal repeat embedded in the middle of the NSD1 gene is actually transcriptionally active and producing more RNA than the canonical gene that initiates at the 5' end of this gene. And so this gives you a sense that in growing oocytes and fully grown oocytes, the ratio of this truncated LTR transcript to the canonical, and the truncated transcript, if it produces a polypeptide, which we don't know because we don't have a good antibody to test this, would lose one of the PWWP domains. And so we don't know if this is indeed produced, it may poison the canonical or full-length polypeptide that's produced. But we think that it is likely the case that NSD1 is at the protein level is either not expressed or not functional because of this very low level of K36 dimethylation independent of SETD2 that we observe in the female germline.
Okay, thanks, Matt. Next question is, are there changes in H3K27 trimethyl or H3K4 trimethyl distribution in the SETD2 knockout oocytes? Yes.
So H3K4 trimethylation does not change much. I did not have time to show you, and this was work actually that Wei's lab did and is shown in the Nature Genetics paper in 2019, that as in the male germline, there is spreading of K27 trimethylation into gene bodies as well as some promoters of genes that are downregulated in that knockout. So in both male and female germline, the decrease of K36 methylation states, di or trimethyl, leads to aberrant encroachment of K27 trimethylation into regions that previously were not marked with this repressive mark, and a subset of genes that seem to be silenced as a consequence of that spreading.
Okay, good. Next question is about the SETD2 work. And so does that mean that there's much more transcription in females versus male germ cells?
Excellent question. We think so. And the reason for that, I think, at least in part, leads back to this highly active retrotransposon repertoire that we observe in growing oocytes. So there are many LTRs driving transcripts, and these are Pol2 driven, and we see de novo methylation being deposited in these transcription units. But I should also say that there are also very many cryptic transcription start sites in oocytes for RNA Pol2 that do not seem to initiate in an LTR, at least in an annotated LTR. And these also generate domains of K36 trimethylation and in turn de novo DNA methylation. So I think that this does, the K36 tri levels between male and female germlines the difference may be is due at least in part or predominantly to transcription initiating and non-canonical TSSs creating domains of K36 tri and in turn de novo methylation of those regions.
Okay, good. So William Gibson said, great talk, Matt. Is there any equivalence between the human CPG sites flagged by episignatures in humans, e.g. Soto syndrome, and the orthologous sites in mice?
That's a good question. We have not directly compared the methylome data from Soto syndrome, which I should point out is microarray based. So as far as I know, there is not whole genome bisulfite sequencing data from Soto's patients. But if there were, we would be comparing blood, most likely, to germ cell methylation levels. And of course, the DNA methylation levels are quite different in wild type due to differences in the transcriptome and chromatin structure. So I think we would probably not expect to see a super high correlation between blood cell, between NSD1 deficient blood cell and NSD1 deficient male germ cell profiles. With the key point being that in the male germline, essentially the entire genome is DNA methylated. I think it may be the most methylated cell type in mammals, with only H3K4 marked promoters lacking DNA methylation, and essentially the rest of the genome at 100% or close to 100% methylation.
Okay, good. And I will ask the last question. So when we're looking at the H3 K36 di and trimethyl across the genome, it tends to be excluded from TSS sites, and the image on the screen there showing that you don't have DNA methylation at the TSS. Have you looked at the DNA polymerase? And do you get transcriptional pausing at these genes here, and is transcriptional pausing lost in the NSD1 mouse?
Good question. We have not looked at transcriptional pausing. One possible explanation, as you can see in the slide, I didn't mention it, but here's H3K4 trimethylation that sort of sits in the hole of H3K36 di and trimethylation. And we know that there's several CXXC proteins, so these are proteins that would bind to unmethylated, in particular CPG island promoters, have H3K36 demethylase activity. So these KDMs. So the lack of K36 methylation in these regions may well be due to recruitment of K36 demethylases to these promoter regions, but we haven't checked what the consequences are in terms of RNA Pol2 release when K36 methylation levels are decreased downstream of the transcription start site.